How to Master MCAT Bio, Biochem pH and Buffering Systems. Are you ready to tackle the tricky world of pH and buffering systems for the medical college admission test biology and biochemistry section? Understanding these concepts is essential for your success. Let's break it down step by step. First, grasp the basics of pH. pH is a measure of hydrogen ion concentration in a solution. It tells us how acidic or basic something is, with a scale ranging from 0 to 14. A lower pH means more acidity, while a higher pH indicates more basicity. Acids increase hydrogen ion concentration, while bases decrease it. Now let's talk about buffers. Buffers are special solutions that resist changes in pH when small amounts of acids or bases are added. They usually consist of a weak acid and its conjugate base or a weak base and its conjugate acid. This is where the magic happens. Buffers maintain pH stability through neutralization reactions. When you add an acid, the conjugate base in the buffer reacts with the hydrogen ions to form the weak acid, which minimizes any change in pH. On the flip side, when you add a base, the weak acid donates a proton to neutralize it, again keeping the pH stable. This is vital for biological systems, especially in maintaining the right pH for enzyme function. Next, familiarize yourself with the Henderson-Hasselbalch equation. This equation connects pH, the acid dissociation constant, and the ratio of conjugate base to acid concentrations. The equation looks like this. pH equals pKa plus the logarithm of the concentration of the conjugate base divided by the concentration of the weak acid. Practice using this equation with various buffer systems, especially biological ones. A great example is the bicarbonate-carbonic acid system, which keeps blood pH around 7.4. This balance is essential for overall homeostasis. Understanding buffer capacity is also important. Buffer capacity refers to how well a buffer can resist pH changes. It depends on the concentrations of the acid and conjugate base. A buffer is most effective when the concentrations are roughly equal, around a 1-to-1 ratio. Buffers work best within a pH range of about 1 unit above and below the pKa of the weak acid. To solidify your knowledge, work on practice problems that involve titrations. Titration curves show how the pH changes as you add strong acids or bases to weak acid or base pairs. Pay attention to the equivalence point, where all the weak acid has been converted to its conjugate base. This point significantly affects pH. From a medical school perspective, it's crucial to connect these concepts to physiological processes. For instance, blood buffers like bicarbonate, phosphate, and protein buffers play a key role in maintaining acid-base balance. Disorders such as acidosis and alkalosis arise from disruptions in these systems, making it essential to master these ideas for clinical applications. To sum it up, mastering pH and buffering systems for the medical college admission test involves understanding acid-base fundamentals, buffer composition, and neutralization reactions. Learn and apply the Henderson-Hasselbalch equation, study buffer capacity, and practice titration problems. Finally, link these concepts to physiological processes and clinical implications. Consistent practice will build your confidence in this topic and help you excel on the test.